Hello students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about genetic code. So at first, what is genetic code? So we all know that in our chromosome, which is the DNA, on DNA many nucleotides are present. Okay, in a specific sequence like G, T, T, A, C, C, G, T, A. During protein synthesis, this DNA sequence is converted into mRNA. A particular DNA sequence is converted into mRNA, form mRNA by the process of transcription inside the nucleus. Then mRNA enter into the cytoplasm outside the nucleus to form a specific protein by the process of translation. So how this message encoded in DNA is transformed into protein? So basically it is due to the genetic code, because genetic codes are present on DNA which form specific amino acid, and those amino acid join to form the linear sequence of protein. So the genetic code is the set of rules used by living cells or set of nucleotides present in living cells to translate information encoded within genetic material into protein. It translates a 4-lettered nucleic acid language into 20-lettered amino acid language. So basically it is a translation process from nucleic acid language to protein language or amino acid language. There are 4 nucleic acids and there are 20 different types of amino acids. So this process, this genetic code helps to translate 4-lettered nucleic acid into 20-lettered amino acid. They specify how nucleotide sequence of mRNA is translated into amino acids to form polypeptide chain by the process of translation. The relationship between nucleotide sequence of mRNA and amino acid sequence of polypeptide is the genetic code. Okay, because genetic code is the secret message and it codes for a specific amino acid. Okay. So these genetic codes are sequence of nucleotides, or more specifically these are sequence of nitrogenous base, either adenine or guanine or uracil or cytosine, present on mRNA. They originate from DNA. Their bases are complementary to one segment of DNA from where they are originated. Okay. It plays very key role or significant role in arranging the amino acid sequences in polypeptide chain or linear chain of protein during protein synthesis. So how to arrange amino acid? At first, Gamow in 1951 first raised a concept of genetic code which can be triplet. That means 3 bases combinedly code for one amino acid. Okay. So what are the key properties of genetic code? At first, it should be triplet in nature. AUG, GUU, UUA, like this. There are 64 possible codons, easily matching the 20 required amino acids. Next key property is the degeneracy or redundant. multiple codons can code for same amino acids. For example, 6 different codons code for leucine amino acid. Out of 64 codons, 61 for amino acids, 3 serves as punctuation marks. Then next property, non-ambiguous, that means specific. Each specific codon codes for only one amino acid. For example, AUG always codes for methionine. AUG never codes for leucine. So it is very, very specific. Non-overlapping, the genetic code reads sequentially 3 bases at one time. A single nucleotide cannot be a part of 2 adjacent codon. Okay. Commaless, it should be commaless AUG. There is no comma in between these 3 letters. Nearly universal, same codon, the same codon code for same amino acids across virtually all life forms, from bacteria to human. Minor exceptions are there, but nearly all amino acids code for same, nearly all codons code for same amino acid. Polarity, the molecular machinery always reads the code in a fixed direction from 5' end to 3' end of mRNA. mRNA, suppose the mRNA is like that, 5' to 3' U U G A U G. So it is the starting codon. So this molecular machinery always reads the code like this, from this to this direction. So this table showing different combinations, how different types of amino acids can be formed from 4 different types of nucleotides. There are 4 types of nucleotides but 20 different types of amino acids. So those nucleotides combine in different forms to form different amino acids. Okay. Next, there are some special type of codons like chain initiation codon, AUG, and sometimes GUG. AUG always codes for methionine and GUG codes for valine respectively. So they always regarded as the starting codon or chain initiation codon. That means a chain or polypeptide chain always initiate with methionine. Next, chain termination codon, that is a signal to terminate the protein synthesis. Here there are 3 different types of chain stop codon or chain termination codon like UAA, UAG, UGA. They do not code for any amino acids. When any of them occurs immediately after the triplet AUG. AUG is the starting codon, then protein synthesis starts, and when one of these appear, it causes the release of polypeptide chain from the ribosome, and it is also called stop codon. They can also called as chain termination codons because these codons are used by the cell to signal the natural end of translation. Okay. Then sense codon. What is sense codon? Sense codon means 61 codons which code for particular amino acids are termed as sense codon. Nonsense codon, UAA, UGA, UAG, these are called they cannot code for any amino acid. So they were originally described as nonsense codon. However, so-called nonsense codons have now been found to be special sense. These special sense codons perform the function of punctuating genetic messages like full stop at the end of the sentence. So it acts as a full stop of that complete polypeptide chain. So they are also called chain termination codon, sometimes nonsense codon, and sometimes stop codon. Okay. So these are some special type of codons. So with this I am concluding my lecture video here on the topic genetic code. I hope you will get benefit from this. This is a very basic video on genetic code. So in my next video I will share some more important topics of biology, or more specifically on botany. So please stay tuned. If you liked this video, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.